So, welcome, students. Happy Wednesday. I believe graduation is happening a little bit later today. Congratulations if that is you. How exciting! Thank you for being with us today. Um, again, my name is Jamaica Marshall, and I am the Shadow the Scientist Programs Coordinator. And I am grateful to be able to connect you, students, and the public in general with amazing scientists as they conduct research and activities related to that research virtually in real time so that you can not only see what it's like to be in a lab or to be out in the field, but ask questions and see how they interact with each other to solve problems. And of course, the research aspect is always just so fascinating. And you will not be disappointed. You will be with Jessica Bonnar and Mikala Capage today. And it's going to be Mikala who is really leading this first pre-session. So how about we go over to Mikala to get us started?
Okay, thank you so much for the introduction. So yeah, my name is Mikala. Before we jump into the science, I'm going to tell you just a little bit about myself. So I'm from Portland, Oregon, out on the West Coast. I was raised in Portland, and then I went to undergrad at the University of Oregon in Eugene. Go Ducks. And then after I graduated with my bachelor's degree in biology, I started at the MIT biology PhD program, and I joined Ruth Lehman's lab at the Whitehead Institute. And so this is a picture of the building where I work. That's actually where Jess and I are located right now. And as a scientist, I'm really interested in the process of development and asking questions about developmental biology. And really broadly, like developmental biology is the process of how you go from a single cell zygote to a multicellular organism. And so that's kind of the broadest question that I study in Ruth's lab. And we use the model organism Drosophila melanogaster, or just the common fruit fly, as the model to answer our scientific questions. And I also added a picture of a microscope to the slide because I do a lot of microscopy using, multiple different types of microscopes and really using, microscopy like every day in my science. So now I'm going to tell you a little bit more about the big scientific concept that we're going to start the presentation off with.
And real quick, also, I just want to say hi. I can't see everyone again, but we're really excited. I was in your class, I think, in December, so nice to be here again. We also really want to emphasize throughout this presentation, please ask questions. We will continue to make time and leave space and ask for you to have questions. We got some amazing questions from first period. I have my eye trained on Mr. Woodbury, so whenever he starts to wave at the screen, we'll pause and take anything. But please, please, please ask questions. There really are no wrong questions here.
Yeah. Awesome. Thanks, Jess.
Yeah.
So the big scientific concept that I want to start off with is the idea of a stem cell. And I am really interested as a scientist in stem cells because these are really powerful cells in your body because they have the potential to become many different types of cells. So what I have diagrammed on the screen is, sort of that concept in visual form, which is that a stem cell has the capacity to become a cell in your nervous system, like a nerve, or a cell in the rest of your body, like a muscle cell, a skin cell, or a fat cell. And I'm really particularly focused on a type of stem cells that are called egg stem cells. And these cells divide to produce the eggs of Drosophila melanogaster. And so the egg stem cells will divide, they'll create an egg, that egg gets fertilized and then can develop into a whole new Drosophila fly. And so these egg stem cells are really powerful because they are kind of the key to the next generation. Without egg stem cells, fruit flies can't lay eggs, they can't make the next generation. So these are like really, really important stem cells. I want to talk a little bit about where stem cells are located in the adult fly. So this is a diagram of an adult female fly, and the majority of her abdomen is actually taken up by 2 ovaries. And the ovaries here are diagrammed in part B of this figure. And at the very, very, very tip of each ovary is this specialized compartment shown in part C of this figure. And then at the very tip of the specialized compartment shown in orange, those are the egg stem cells. And so the egg stem cells reside in the fly ovary at the very tip of the ovary, which is housed in the abdomen of the fly. So that's where egg stem cells are located in the adult. But before the fly is an adult fly, egg stem cells are actually developing in the larval fly ovary. And so this is an image that I took using one of our microscopes, but this is an image of every single cell in the larval fly ovary. And what you're looking at here in the image is just the DNA content of the cells. So basically the DNA that's inside every single nucleus inside each cell is just marked in gray. And so you can kind of see all of the different cells that make up the ovary. And the goal of my experiments is to count the number of egg stem cells. But if I just show you this image, just looking at the DNA of the cells, you can't really learn very much about the egg stem cells. For example, I don't know in this image where the egg stem cells are or how many of them there are. And so essentially, like, it's interesting to look at all of the DNA in the cells, but that's like not the most informative way of looking at the organ. And so what I need to do is to perform a type of scientific protocol called an immunofluorescence protocol. And immunofluorescence protocols identify egg stem cells. And so on the left here is an image of what that fly ovary looks like without doing the immunofluorescence. And then on the right is what the image would look like after doing the immunofluorescence protocol. And all of a sudden, there are now cells marked in magenta, in this like bright pink color, and those are the egg stem cells. So that sort of wraps up part A of the handout that you all are working through. And what I'm going to be jumping into next is actually going through kind of a week in the life in the lab, what it's like for me to do this immunofluorescence protocol from start to finish. But before we move into part B, I just want to stop and make sure and ask if there are any questions about stem cells or egg stem cells in particular.
You guys got any questions? I think we're good.
Okay.
Are there any blanks in part A that students want us to go over? Maybe they didn't quite grab it.
No, I think you got it. I see a couple head nods that we're all good.
Okay.
All right.
So the protocol that I'm going to be working through that I've been doing over the last couple days is again called this immunofluorescence protocol. And I do this protocol over 4 days. And so on day 1, it starts out with the process of dissecting larvae. And so I'm going to talk a little bit about that. These are some images of Drosophila larvae in the lab. And really quickly, I also want to just show, this is what the life cycle of Drosophila is. So you go from, like I said, this adult female fly that will lay an egg. That egg hatches into a larvae, which then grows bigger and bigger and bigger. And then because these are insects, what they will also do is crawl up like the side of, somewhere in their environment, and they'll form a pupae. And then they hatch out of that pupae as like a, adult fly with wings. And they're sort of capable of starting this life cycle over again. And so the egg stem cells live in the female fly, but I'm interested in studying the number of egg stem cells in these larval stages of development. And so I'm actually not even looking at the adult, I'm looking at this earlier developmental stage. And so this is what the larvae look like in their little vials. They live in vials of food, so they sort of live and eat at the same time. And then these vials are plugged with a little cotton cap to sort of keep the larvae in. And this is what these Drosophila larvae look like under the microscope. In order to dissect the tissue, I use these like very small tweezers, and I have to do these dissections under the microscope because it's too small to kind of see without some magnification. So the very first thing that I do is I take the larvae, I work with them under the scope to dissect out their tissues. And then this is what the dissected tissue looks like in these little tubes. So after I dissect it out of the larvae, I place the tissue in this tube of liquid, and the tissue will stay in these little tubes throughout the rest of the immunofluorescence protocol until I'm ready to put them on the microscope. So that's kind of day one of the immunofluorescence protocol. It basically starts with like acquiring the organs where the egg stem cells are located. So any questions about larvae or larvae in tubes, the process of dissecting? Flies in general?
You guys got any questions? I'm just trying to think about the questions that we had in period one. I think one of them was like, obviously, since, you know, these are larvae that you're working with, you have to use a microscope in order to do dissections of these. And so in period one, I was kind of telling students like, oh yeah, when, you know, you're using a microscope, like any movement of your hand is just amplified. So if you drink like caffeine or coffee before you go in a lab and do these dissections or working with a microscope, your instrument, your tools, your probes, whatever you're using, it's just kind of going all over the place on the screen. So having like really delicate hands and everything, certainly a really good skill. So nice surgical hands. And then yeah, did you have a question? Yeah.
So I know like the whole goal of this experiment is to count the number of egg stem cells, but I guess like after you accomplish that, like you know how many stem cells you're working with, I guess like what are you going to do with that information in the future? Like how is that going to help I guess further scientific research after this?
Yeah,
Excellent.
a fantastic question. And I feel like that question really highlights that something that's very important. Like the goal is not just to know, the goal is not to necessarily just know how many egg stem cells there are, but actually understand what are the developmental mechanisms that control how many egg stem cells there are. And so what I mean by that is that the number of egg stem cells is determined by lots of different like factors and pathways that are acting during development, but we don't have a complete understanding of what controls the number of these cells. And so what I want to do is I want to actually understand if I break something in the developmental process, will that change how many egg stem cells there are? And so what I do is actually do control experiments. And so I have control samples and experimental samples. And in my control sample, they're just, you know, normal, wild type. Nothing is like changed in their like developmental systems. And I understand how many egg stem cells are in that population. And then I have experimental samples where I've deleted a gene or I've changed you know, how a gene functions or where it functions or when it functions. And then what I'm looking for is for changes in the number of egg stem cells. And if I see a change in the number of egg stem cells, either fewer or maybe more egg stem cells, that means that that gene that I disrupted contributes to egg stem cell growth. And so it's really a process of breaking something in the developmental system and seeing if you can see a change, because then that tells you that that thing you broke was important, basically.
It's a great question, and like the process that Mikala just described is not a process that is specific to thinking about ovary organ development. It's a core principle that all biologists use. I do structural biology and I do the same thing, where you break something and you look at the change, and if there is a change, you know that what you've broken matters.
Yeah.
Kind of, you don't know what you've got till it's gone.
Yeah,
right?
Exactly.
Would you mind just like briefly saying what you're breaking tomorrow?
So in between, so in the experiment that we're going to be imaging together tomorrow, I have deleted one very particular gene in the larval ovary. And that gene is important for the ability of cells to contact each other. And contact each other and like form very strong connections between two cells. So the cells basically express this protein on their membrane, and the protein allows the cells to contact each other. And I've deleted that protein. And so what I'm looking for is to see if getting rid of this cell contact protein is important for controlling the number of egg stem cells. And I'll tell you that the protein, I'll tell you the name, and I'm actually testing a lot of different proteins tomorrow, but the one that I think I'll be imaging with you is called coracle. Fun name.
Thank you for the question. It's
Yeah.
a great, great question.
All right, and we got another question too. What's up?
So flies are your model organism because this protein is universal, or is it because they reproduce fast?
Sorry, I missed, I think, just one part of that. Would you repeat it?
Yeah, the question was, is this protein found in fruit flies, is this a universal protein or is just specific to the fruit flies?
Yeah, it's a really good question. So here Drosophila is serving as my model organism. I'm using it as a system that will let me sort of like rapidly test my ideas. And that's because Drosophila grow very, very fast. They're very cheap to maintain and work with. And so they're a really good organism for trying like a million ideas and seeing which ones are good ideas. And I will say too that the proteins that I'm interested in, they're not universal to humans, but humans have very similar proteins. And so the scientific word that we would use here is that humans have homologs of these Drosophila proteins. So essentially, they're not the exact same, but there are definitely similar ones in humans. And the goal would be that what I find in Drosophila is then applicable to how this process works in humans.
Cool. Any other questions that you guys have? Yeah, those are good questions so far, so let me know when you got them.
Thank you.
Yeah. Okay, so some great questions about like, why am I doing this and why am I doing this with Drosophila? So in order to, again, understand how the number of egg stem cells is controlled, I first have to understand how many egg stem cells there are. And to understand how many egg stem cells there are, I have to be able to see them. And so that is what this immunofluorescence protocol is letting me do. So on the first day, or on the second day of the immunofluorescence protocol, after I have my dissected tissues, I add a primary antibody. And primary antibodies will help me identify egg stem cells. And the way that they do this is based immunofluorescence assays are identifying cells based on the proteins that the cells already contain. And so this is kind of a very like fundamental concept to this protocol, which is that cells that are different types will contain different proteins naturally. And we as researchers can take advantage of that by identifying those particular proteins and using that as a way to identify different cells. And so the protein that I want to introduce to you all is called the VASA protein. And what we know is that egg stem cells contain vasa, and non-stem cells do not contain vasa. And Drosophila are really, really good at dividing their cells like really faithfully. So only egg stem cells will have vasa, and non-stem cells like do not even contain a drop of vasa. Like they are really, really good at making sure that vasa is only in egg stem cells. And so basically, in reality, there's this underlying difference that egg stem cells contain vasa, but I don't know where the vasa protein is. And so that's where this primary antibody comes into play. So primary antibody, the vasa primary antibody will only bind to the vasa protein. So I take my tissue and I add a little drop of a primary antibody. And this is what these primary antibodies look like in the lab. They come in these little tubes, and you can see that they're like labeled with different names. So one of them is the vasa antibody. And so it's basically just a little drop of liquid. And what's happening when I add that liquid to the tube is that the primary antibody is only binding to the vasa protein. So now my egg stem cells are going to have primary antibody, and my non-stem cells don't have any antibody. So if I were to image my samples after just adding just primary antibody, it would actually still look like this. It wouldn't look any different from the beginning of the experiment because I actually can't see the primary antibody. So all I've done so far is kick my problem one step down the road. I have identified a difference. I've added primary antibody, but I can't see the primary antibody. So I need to do one more thing. And so I finish out day 2. I come back on day 3, and on day 3, I'm going to add something called a secondary antibody. Before I jump into that, I want to ask, are there any questions about primary antibodies?
You guys got any questions? I think one of the questions from period 1 was, and this is one that I asked, is like, where or how do you get the antibodies that bind to vasa? Yeah,
a fantastic question. So, oops, no. Don't look. Yeah, the question of where antibodies come from is a really fantastic question, and I'm going to introduce an idea that is going to come out of left field a little bit, but I promise that it is connected. So I don't know if anyone has ever told you when you're sick, if you have like a virus or some sort of infection, if anyone's ever said like, oh, your immune system is like fighting off this infection. That's why when you're sick, you sometimes run a fever because your body has sensed that there's an invading protein in the form of a virus, and it's doing everything it can to get rid of that infection. Scientists have taken advantage. And so what your body is doing when you're fighting off an infection with your immune system is your body is producing antibodies against the foreign proteins. And so scientists have taken advantage of the fact that an animal has an immune system to basically force an animal's immune system to develop antibodies against specific proteins. So the vasa antibody is made by introducing purified vasa protein into a research animal and letting that animal's immune system create antibodies against that vasa protein.
Yeah, and I will say I have worked with mice in the past, so a lot of antibodies are commonly made in mice and rabbits. There's sometimes other research animals that they're made in. But one of the things that I just want to point out is like how ingrained in working with animals it is important to respect the animal and care for the animal. I didn't mention this in first period, but it is actually like federal law that you treat the animals with respect. And if you have plans to like cause pain, or you think that animals might have pain caused during a procedure, you have to go through a very rigorous set of steps to get that experimental protocol approved and show that you're doing everything in your power to keep the animal comfortable. I will also say the mice that I worked with, like whenever we sacrificed the mice, we always thanked them for their sacrifice and for bettering science. But fundamentally, like to understand these really fundamental things, we do need reagents that come from animals and come from their immune system. So I just wanted to mention that. And also, I think I mentioned this in first period as well, but these vials for Mikala's are not super expensive, in part because they're vials that your lab themselves have generated. But a lot of vials of antibodies, like this vial of antibodies that's on the right, can cost upwards of $500 a vial, which I know is expensive. But if you think about it, like this comes from like taking really good care of rabbits and like feeding them and making and like purifying protein, and also the time and expertise required to understand how to properly purify the protein. So, right, this little vial like doesn't seem exciting.
Yeah, but it represents like I think it's very cool because it is both a tool to do future research, and it represents a really deep understanding of how animals already work, right? Like there had to be research that went into understanding how to make an antibody, and now that antibody is serving as a tool for future research.
Yeah.
So like everything that I do in the lab is the result of like lots and lots of people's work in who came before me. And even particularly in the case of this Vaza antibody, this was made by my lab like 30 years ago. And we have been sending out this antibody to researchers all over the world for the past like 30 years. And so it's very, very precious. It's a very precious resource. And so I use very, very small amounts and I try not to waste it. But it really is the result of a lot of work that came before me. And now I get to use it to make new discoveries as well.
Yeah. So we're basically standing on the shoulders of giants to see
Yeah.
better than they can. That's like a common theme in science. You guys all understand.
Very interdisciplinary. And I should say, Mikala just explained the extent of her knowledge of immunology.
Yeah, that's all
that
I know.
Our two brains have together.
Yeah, please
don't ask more.
Don't ask more. We love questions, but not about immunology.
No, that's all we can give. Any other questions?
Yeah, we got a question.
I don't know if it's, but
yeah.
How did you get the antibodies to last so long, like 30 years?
Yeah. The question is, did you hear that question?
How do you get them to like last that long?
Yeah, okay.
Yeah. Part of it is that part of it really just comes down to like the amount of antibody that you get from an animal and the amount that you need for your experiment. So from when you produce an antibody, you could get like a liter maybe of antibody. And I wish I had so you could get like about a liter of like pure antibody that's in this tube. And then for my experiments, I use I can't even explain how
one millionth.
Yeah, 1 × 10⁻10th liters.
Yeah, 1/10 billionth of that is used for experiments. So you can do 10 billion experiments.
Yeah. And then you can also reuse the liquid. So I will often add the liquid to a sample, let it sit we call it incubating for about like overnight. And then I can actually pull that liquid off and use it again. So between a lot of initial volume, using a very small volume, and reusing, that's how you can get it to like last a good long time.
I'll add, that's a super interesting answer. I was thinking of the question in a very different way, which is, the antibody is fundamentally a protein, and how are you able to get a protein to stick around for 30 years and just to continue to exist for 30 years? And so the other thing that I'll just say is like, this comes from a really fundamental understanding of biochemistry and how proteins fold. And so the solution that this protein is stored in is a huge part of it is glycerol, which we know is really helpful. Well, a lot of times it's glycerol for antibodies, maybe not this one, but like basically protein biochemists have studied antibodies for a long time and also know like how to store them in liquid that helps them stick around for a long time and helps keep the proteins folded as well. So again, multiple ways that science comes and helps create this little tube.
Yeah, good question.
Yeah.
Cool. And I think we got another question over here.
Right.
Okay, I know this is kind of going ahead, but you talking about like the cost of primary antibodies made me think of this, and I'm just curious if you put like the dye on the secondary antibodies for imaging, if that's because like secondary antibodies are like cheaper to produce or
Yes.
okay.
Yeah,
That's
so
exactly it. Yeah.
oh my gosh. Okay, so this is also kind of a really cool like innovation that came from like antibody research, I guess. So yeah, so the way that we're jumping ahead a little bit, but yeah,
Maybe
okay.
talk about secondary and
I'm
then
gonna,
she'll answer your
I'm gonna
question.
talk about secondary and then I'm gonna answer this question.
And
so
you're correct.
yes. So okay, so we added our primary antibody. We didn't make any progress. We still can't see the primary antibody. We move on, we look at a secondary antibody. And essentially, a secondary antibody is specialized and will only bind to certain primary antibodies. And so the secondary antibody is attached to a fluorescent dye. And basically, the situation that I have before adding the secondary antibody is that my egg stem cells have VASA protein. That VASA protein is bound to a primary antibody. And the secondary will now bind to the primary and basically light up the egg stem cells. Whereas the non-stem cells, they never had any VASA protein, they never got any VASA primary antibody, and so they didn't get any secondary antibody, and they don't have any dye. And so this is what the real life secondary antibodies look like in the lab. These liquids are a little more fun because they're bright colors. But again, I'm adding just a very, very small volume of each secondary antibody to the tube, or one secondary antibody to the tube. And the secondary antibody has now lit up the VASA protein. And so now I can tell the difference between an egg stem cell and a non-egg stem cell because the pink is marking the VASA protein. And so if you look at this like inset image that's just taken from the bigger image, you can see there's, a cell that has pink in the cytoplasm, and right next to it is, a cell that doesn't have any pink, and that's not an egg stem cell. So to recap, we have our proteins of interest that mark a specific cell type. We have primary antibodies that are specialized to recognize particular proteins. And then we use secondary antibodies that bind to the primary and are also bound to a dye. And I think the question, oh wait, I got really excited. So what was the question?
I have it. It
is,
do you have the secondary antibody with a dye attached because that helps make things cheaper? Is
that
correct?
Was that, yeah?
Yeah, that's correct.
Yeah. So basically what scientists have figured out is that sometimes you want to look at a particular protein, but you want to look at it in maybe multiple different colors. Sometimes you want, sometimes it's more useful for the experiment for VASA to be in pink, but maybe for other experiments you need it to be in green or in blue. And if your fluorescent dye was attached to the primary antibody directly, you would have to have a VASA antibody that is pink and a VASA antibody that is blue and a VASA antibody that is green. And so then that's 3 antibodies, and that's way more money and way more time. And so what we do instead is have sort of a base primary antibody that'll recognize VASA, and then I can add on these like different color adapters basically, so that one antibody can be either pink or blue or green, et cetera, et cetera. And so that makes the experiments more flexible and cheaper, basically.
Yeah. Could I add one thing on?
Yeah.
In addition, like the primary antibodies, right? You might imagine the VASA protein is not the only protein scientists care about. And in fact, like we've generated primary antibodies against a lot of different proteins. And I mentioned that primary antibodies are often made in rabbits and mice. And I pointed out, or in this diagram here, I noted that like the secondary antibody recognizes something specific on the primary antibody, but it's recognizing the fact that the primary antibody was made in mouse or rabbits. And so what you can do with a secondary antibody is you generate the secondary antibodies such that they recognize any primary antibody made in mouse or rabbits, and then you can attach a dye. And what that means is the secondary antibodies are quite generalizable. So not only is this a secondary antibody that recognizes this specific primary antibody, but it recognizes many, which allows for you to change between experiments. And my point that I'm making with that is that the dye conjugation is an expensive technique. And so also, if you had to conjugate dyes to every single primary antibody that you made, Mikala noted, on one hand, you'd have to conjugate dyes every single time you want to change the color of your primary antibody, but also like it means you don't have to conjugate dye to your primary antibody, period, and you can just buy the secondary antibody that producers mass make, and because they're making so much of the secondary antibody, it's cheaper for them.
Yeah.
I think I maybe made things more complicated, but the answer is like, yes, it helps makes things cheaper. And I think the second point that Mikala was making is it helps make the primary antibody a lot more flexible to not have the dye connected to it.
Yeah. So I wanted to go back actually to this image of these brightly colored tubes, and it's a little bit hard, like it was kind of hard to take the picture, but if you look at the middle tube in particular, it actually says mouse on it. And that is because this secondary antibody is designed for mouse antibodies. So the pink liquid, the antibodies inside this tube will recognize any antibody that came from a mouse. Similarly, the blue tube on the left says rabbit on it, and that is because it will recognize any antibody that was made in a rabbit. And the third one here in green is actually, it doesn't say, you can't see it, but it's from a guinea pig. And so you have like all of these different animals, and then you're doing this like mix and match thing to be able to look at lots of different proteins all at once, very like flexibly and interchangeably.
And I think we got another question too.
So because now your secondary antibodies, they only bind to primary ones from certain types of animals, I guess, could you use a secondary antibody to maybe detect or keep track of any discrepancies that would come from an antibody from a mouse versus an antibody from a rabbit?
I think so, yeah.
Yeah. Do you want to answer that?
So just to check, could you use your secondary antibodies like to sort of like keep track of where your antibodies were made?
Yeah.
Is that the question?
Well, like to keep track, but also like, I mean, I'm not 100% on like how
Sure.
antibody collection works, but I'm just like, if there was any issues of how it was collected or if something's not acting right, you would know exactly which antibody is like acting up.
Yeah,
I see what you're
totally.
saying. Well, and maybe
yes.
just to add on to this, right, you also have to purify the primary antibodies that you're harvesting from a mouse. And I don't know exactly how that purification process works, but it might in fact be that they're purifying the primary antibody, in fact, using this sort of secondary antibody, like extracting it from the rabbit or the mouse in the first place. I actually do techniques similar to that as a biochemist all the time. Not quite answering your question, but if you had other.
Yeah, but basically like this is a big concept in biology, and researchers take advantage of it all the time, which is that proteins have a lot of like binding specificity. So some proteins are really good at binding very specifically to some proteins and not others. And so the fact that a mouse antibody can be recognized by like a mouse secondary antibody is an example of protein binding being used as a tool. And proteins are like really good at knowing what it should bind to and not binding to other things.
Yeah.
And we take advantage of that all the time in our science.
And I will say, I won't get into it, but I'm actually using something called a nanobody, which is an antibody that was originally purified from a llama because they have really weird antibodies. And I'm actually using it as a way to see how a specific machinery on the edge of the nucleus is assembling and disassembling. And so like that antibody, I am using it to light up, but I'm actually using it to like give me an understanding of if the machinery is broken or not. So just expanding upon this idea of like proteins recognizing and binding protein surfaces, it's like another really core concept in biology.
And we got another question.
Cool.
So the fluorescent dye, is it synthetic or is it produced with certain bacteria? Because, like you said, the secondary antibody, I know it will bind to the specific animal that it came from, but like you said, proteins are very specific with their binding. So how is that fluorescent dye attaching to the secondary antibody?
Great question, and this is where we get into fundamental chemistry. So I know more about fluorescent dyes than I should because I had to take an organic synthesis chemistry class. But basically, these the fluorescent dyes, sorry, are artificially created by chemists. I do just want to point out these are labeled as alexifluor dyes, right? So these secondary antibodies are conjugated to a dye called an alexifluor. It's a rhodamine dye, which is a very specific chemical structure where if you ever end up taking organic chemistry, you'll sort of learn about the concept of resonance or of electrons moving around an electron ring.
Okay.
Sorry, no, I'll talk about it. Yeah, great question. But like basically chemists have thought very carefully about some of these things. There's someone named Luke Lavis, who like loves reading about this. And if you're interested in learning more, he has some great reviews with very funny names. But these are alexifluor dyes, and he's invented geneliifluor dyes, which are actually just as bright as alexifluor dyes, and he's really interested in sharing them broadly with the scientific community.
Yeah, but
So-
basically,
Yes.
you're definitely right in that like part of what biologists do in the lab is we take advantage of what nature has already come up with, and then we add in some like chemistry and some synthetic chemistry to kind of like make it even better.
And so to close that loop, these dyes that are then synthesized, chemists are really, really good at understanding how to attach a small molecule to a protein. And so basically you have chemical synthesis where this dye has been made, and then the dye gets attached to the antibody that is purified.
Yeah.
And that is specifically what happens. And I'm sorry if I went off on a tangent.
And so finally, like with this idea of like standing on the shoulders of giants in the labs that Jess and I are in, it's not just that we're relying on everything that people know and have learned about biology, but we are also taking advantage of things that people know about chemistry and about physics and about material science and about ecology, and like all the different scientific knowledge is all kind of being used to make these experiments work. So, you know, one way that you can think about a microscope, which we'll be using tomorrow together, is just like as a physics question, basically. So like there are physicists who think a lot about how to like use microscopes in the best way possible. And so my experiments are also really possible because of a lot of advancements in physics and chemistry and biology and all of this different stuff.
As one more example of this, my partner works at Boston Scientific, and he is a mechanical engineer by training, but he works on a device that does non-invasive surgeries for heart surgery. And so he uses his mechanical engineering training to help make heart surgeons' lives easier. And so I think one of the things as you continue to progress across science is like, it's really interdisciplinary.
Yeah. Yeah, you can't forget about the engineers.
You can.
Okay, so those were some fantastic questions about, you know, chemical synthesis and secondary antibodies. I want to make sure that we get to the very last part of the worksheet that we all have, which is that, you know, we just keep coming back to this slide, but you'll note that I actually showed you 3 different tubes of secondary antibodies. And that's because when I do these immunofluorescence protocols, I actually am looking for 3 different proteins at the same time. So I'm going to tell you a little bit about what those other proteins are. So we've talked a lot about VASA, which is located in the egg stem cells. But I also want to tell you about the protein traffic jam. Traffic jam in all of our images is going to be green, and traffic jam is in the nucleus of non-stem cells. And so traffic jam is not in the egg stem cells. It just marks the nucleus of non-stem cells. And And the way that I identify traffic jam is about the same as the way I identify VASA, except I'm using a traffic jam specific antibody and a secondary antibody that is conjugated to a green fluorescent dye instead of a pink one. And then finally, we're also going to be looking at the protein HTS. HTS is an interesting example because it's actually located in both egg stem cells and non-stem cells, but in different places in the cell. So in egg stem cells, HTS is localized into a specialized compartment within the cell. And in non-stem cells, HTS is on the membrane. So it basically marks the outline of all of the cells. And so this is sort of an example of a final product image. This is the type of image that we're going to be producing together on Thursday. But basically you have your egg stem cells, which have the pink protein for VASA. It's a little bit hard to see with this overlay, but you can also maybe see, particularly on the right side of the image, a couple of those dots of HTS in red. Then you can also see the red signal marking the outline of all of the cells. And then you have traffic jam in green marking the nucleus of the non-stem cells. And so basically I'm looking at all three of these things together, as well as looking at the DNA inside all of the cells. That's what's in gray. And so it's really like all four pieces of information at once in one image. And so these are the types of images that we're going to be creating and looking at together tomorrow. That's day 4 of the protocol, which is when I image. And I'm really excited to see these samples with you all. So that is everything we have. I do want to throw up, or actually, let's see, what do I want to do? So first I actually want to make sure that we've answered any questions that you have about the handout. At this point, you should be able to fill out the worksheet entirely. So if there are any missing parts or any additional questions, let's do those.
Any missing parts, you guys? Any additional questions? Looks like we're all good on our end here.
All right. So the next thing that we would love is if you would fill out this short survey. This just lets us know if there's anything we want to cover in particular tomorrow. So if you would grab this link to start.
And we can maybe skip.
Yeah.
But if they have any questions that they want to, maybe while they're doing it, we can talk about how we got started inside.
Sure.
I think we got a question here.
Perfect.
Nice.
So I was just wondering, so you're counting number of eggs in the larva. I was just curious if like number of eggs, if it follows like any type of like inheritance pattern of like, if the mother has like 60 eggs, is it more likely that the offspring will have 60 eggs as well?
It's a really good question. Basically like, it doesn't necessarily follow an inheritance pattern, like with in terms of number, but I will say that like the number of egg stem cells is really tightly controlled. So if you're just looking at wild type control samples where I haven't broken anything and I haven't like changed anything about the genes, there tends to be a really like tight range in terms of the number of egg stem cells that an organism has. And in order to develop, it's at the stages that we're looking at, it's about 100. And so the range that I see is somewhere between like 90 and 120. And so the Drosophila knows somehow that it wants to wind up with about 100 egg stem cells, and it doesn't really have too many errors. And so the ability to make 100 egg stem cells consistently is heavily regulated. Basically, there are a ton of different mechanisms that ensure that a Drosophila can make about 100 egg stem cells, knows to make about 100 egg stem cells, won't make fewer, won't make more. And so it's really that very intricate developmental process that I am trying to break.
And to add one thing onto that, thinking back, like the question of like, is the number of egg stem cells the mother have relate to the child, like going back to genetics and sort of like this information being fundamentally encoded in DNA, the model organisms that Mikala are using, correct me if I'm wrong, in terms of the wild type, are genetically identical.
Yeah.
They are genetically identical, right? So like this 100-ish eggs number is set up in part because the flies are genetically identical. And then when you make these changes in the mother, for example, like if you make a genetic change in the mother, or if you make a genetic change in the offspring, that very precise genetic change, because we know the genetics are otherwise identical, like allows for starting to understand those processes in more detail.
Yeah, exactly.
Other questions?
Yeah, we got a question.
Again, why the immunofluorescence binding to the secondary antibody is more efficient and cheaper, and why it would take a third antibody and bind it to the primary antibody.
Would you repeat that for me?
You want to come over here, Corey, to say it a little louder?
Just about like why it would take a third antibody if the immunofluorescence was binding to the primary antibody.
Say that one more time.
One more time.
Sorry.
All good. She's getting a little closer to the microphone on the computer.
Why it would be more efficient and why it would take a third antibody if the immunofluorescence was binded to the primary antibody instead of the secondary antibody.
I think I can maybe re-clarify.
Yeah.
So if in this photo, the immunofluorescence were bound to the primary antibody, you'd actually only need the primary antibody for this given experiment.
It would do like two things at once. It would be able to identify the VASA protein, and it would be able to light up all in one step. And that's kind of nice. Like that does sound kind of faster because, you know, I did say that like doing the secondary antibody does take an extra day. So on the one hand, maybe that does sound nice to combine them, but.
And I will say, I've worked at companies like biotech companies that just have not infinite, but a lot of money to spend on science. And we will buy primary antibodies conjugated to dyes specifically for that reason, to save a day. However, in terms of thinking about the cost of doing science, primary antibodies, there are truly millions of them that exist across the world, and you need a new primary antibody for every protein target of interest. In a given experiment that I might do in the lab, I might have 5 proteins of interest that I want to add a primary antibody to. And all of those primary antibodies need to light up different colors from each other, right? And so whenever you want to change the color of a primary antibody, if it were specifically conjugated to the dye, you would need to buy a new primary antibody conjugated to a new color. However, if we leave the primary antibodies with no color on them and we simply add a secondary antibody on as needed with colors that make sense, it ultimately makes things a lot easier. And also those secondary antibodies in general, because they can be broader, we know they need to recognize a primary antibody made in rabbit, rat, mouse, guinea pig. Companies will make them in huge batches that ultimately makes them cheaper to produce and cheaper to buy.
Yeah. So one other thing I would point out is like the secondary antibodies that I took this picture of, we buy these from a company. And everyone in the lab can use them because they are so generable and interchangeable. But the primary antibodies that I'm using, the tiny one labeled TJGG, that came from a lab in Canada. And then the 1B1 I ordered from a company, and the VASA I got myself. So these are kind of much more kind of like homemade specialty things.
Great question.
Yeah.
Mr. Woodbury said we have less than 1 minute left, so we might finish off.
Yeah, so we're really excited to do some live microscopy with you tomorrow. Thank you for filling out our survey, and we will, same time, same place.
Thank you, Mr. Woodbury, for letting us visit your classroom. Thank you, Jamika, for having Shadow the Scientist and really setting all of this up. And seriously, you all had some amazing questions today. Thank you for your questions, and we're so excited to share with you more tomorrow.
Awesome. Thank you so much, and I'll see you guys tomorrow.
See you tomorrow. Bye-bye.